is my great pleasure that all of these amazing researchers have agreed to take part on our symposium on affective embodiment. And without further ado, I would like to welcome and thank Micah Allen, who's Associate Professor at the Aarhus Institute of Advanced Studies and the Center of Functional Integrative Neuroscience. And Micah has done some very, very interesting and influential work on interoception. And today he will be talking about interoceptive self-inference. And over to you, Micah.
Thanks, Manos. It's really a great pleasure to be here. It was really exciting to get your invite to take part in the symposium. I think I'm not seeing my slides right now, but sorry, everybody, if I do. Yeah, okay. So this is my first time at the Experimental Psychology Society, but it's really an honor to get to present some of our work. I think I read in the conference description that we should present very preliminary work that is not published yet, and that's exactly what I'm going to do today. It's kind of a short two-part talk, with the first part being about some computational modeling we've been doing of interoceptive self-inference, and the second part being about measurement and experimental methods. So I think just to address the overarching theme of effective embodiment and what that kind of means, I think in the context of the overall symposium, is the notion that there's some kind of fundamental link between the way our bodies process internal stimuli and maintain homeostasis and kind of keep us moving through the world, and the affective color or emotional consciousness that we have of ourselves and the world. And that's really what the concept of interoceptive self-inference that I'm going to describe is all about. So normally I would be following from Sarah, who is going to be right after me, and I'm sure going to give a much better overall definition of interoception than I can. But since I'm sort of kicking it off, I'll just get us a little definition, which is when we talk about interoceptive processing or interoception, we're talking about a tradition that starts with, for example, the work of Sherrington, who described the interoceptors as the signals arising from the smooth viscera of the internal organs. And these could be things like signals arising from the heart, the lungs, the stomach, and even slower things like endocrine and metabolic systems. And in general, these are all things that are tied into our homeostasis and our allostasis, the kind of rhythmic, clock-like maintenance of embodiment and living. And we can think about interoception both in terms of the kind of signaling itself arising in the body and what might happen when you have, a cardiac trauma or gastric distress, how that might influence the brain. And we can also think of it from the psychological side in terms of the ability to detect or respond to or control those sensations. And I'm not going to cover it so much, but we often distinguish between explicit interoception, which is your conscious ability to detect those internal changes or signals, versus kind of a more implicit brain-body interaction, which might be some implicit correlation between the rhythm of your heart or the rhythm of your breathing and the way you see an external stimulus. I think we'll probably hear some of that in Sarah's work and perhaps in Manos. So in our current psychological approach to interoception, there's an excellent dimensional approach that's been proposed by Sarah and Hugo Critchley in Sussex, where they distinguish between the basic level of interoceptive accuracy, which is the objective ability to detect visceral sensations, interoceptive sensibility, which is kind of subjective beliefs about interoception, and metacognitive accuracy or interoceptive awareness, which is the correspondence between those three things. And this kind of psychological view of interoception and increased interest in understanding the neural correlates and the mechanisms that underlie each of those three levels is mirrored by a rising awareness in neurology and in psychiatry and neuropsychiatry that deficits in either the bottom-up interoceptive signaling or the more top-down interoceptive control are really transdiagnostic factors that can be seen in a wide variety of different health-harming disorders and psychiatric disorders, very nicely described in this table in a mega review led by Sabib Khalsa and I think 36 other authors, just kind of going down the list and showing how in so many disorders that we typically think of in terms of the cognitive side and the motor side, how there are also somatic visceral disruptions and interoceptive disruptions. So a lot of the interest in interoception and being able to measure it and model it comes from this kind of computational psychiatry or psychiatry-inspired movement. And that's where my group, the Embodied Computation group, is really focused. So we try to blend a computational psychiatry approach with the study of interoception and brain-body interaction. We borrow, for example, ideas from learning theory and decision theory to try to model how signals arising from the interoceptive channel, for example, your heartbeat sensations or the respiratory sensations, might interact with your exteroceptive perception of the world and the decisions you take. The kind of underlying intuition we have here is that interoception is not only useful for monitoring what's happening in your body, but it also is a way that your brain can actually predict and monitor the overall stability or reliability of the sensory epithelium. That is the body as the kit the brain is using to collect the samples. If your heart is palpitating or you're hyperventilating, then the noise level in the brain is going to be elevated in such a way that you may want to adjust the way you make decisions. So we've tried to develop a lot of conceptual and computational models explaining that type of interaction. And we've also recently extended this model to the neurobiological domain, for example, trying to propose a novel model of how the insular cortex, which is very important for processing interoceptive stimuli and is involved in virtually every cognitive domain you can think about. There's actually a meta-analysis by Tagliarcani which showed that the insula is the least specific brain activation region of the whole neocortex. So to put that another way, if you see an insula activation in the task, it gives you very little information about what the task might have been. But nevertheless, we know from rodent studies and electrophysiological stimulation studies that the insula is a very important interoceptive cortex. And we proposed a kind of model you can check out in our recent TICS paper, where you have a gradient from the posterior insula to the anterior insula that is integrating interoceptive and exteroceptive sensations to form higher level metacognitive beliefs about overall levels of expected uncertainty. Now, it's nice to have these kind of conceptual models to make predictions about experimental phenomena and brain data. But we also really think that a big goal in our group, at least, is to develop quantitative computational models that can be used to model interoception itself and also model deficits in psychiatric populations and try to drill down to the level of mechanism. So what I'll briefly present is our first attempt at doing that, where we think about a kind of phenomena that's a little bit outside the usual wheelhouse for interoception, where you're often interested in the direct detection of vagal afferents. So here we ask you to think about something a little different. If you imagine, for example, that at the back of your retina, there at the optic nerve, where the optic nerve enters to the optic disc, there's a large pulsatile vein which actually quivers with each heartbeat. Some people, maybe about 10% of people, can actually see this by pressing on the eyelid or looking in a certain direction. And you can see this here in this positron emission tomography blown up image of the back of the retina, where the retina is being systematically distorted with each heartbeat. So we wanted to build a computational model which would explain this type of cardiac-linked sensory attenuation, which is happening along with the rhythm of the body. Part of our thinking of doing that is thinking in terms of the way the brain combines both predictions about internal signals and confidences or precisions in those signals. So if we kind of look at another example, this is a motion amplified image of a structural brain scan, and we can see that the brain mass is literally quivering with each heartbeat and cardiorespiratory cycle. And we can kind of imagine from the previous example that if you stick an electrode, near one of these pulsatile veins, you're going to have a rhythmic pulse in the noise level of the electrode, which is actually something that was really beautifully shown, I think, in a neuron paper last year, where they showed that the baseline firing rate of neurons is actually being controlled or biased by vasodilation. Now, if you imagine looking at this signal over time, where you have some periodic increase in noise, and then you plot the standard deviation of that noise, you would get a very rhythmic kind of sinusoidal increase and decrease in the noise level or the precision level of the brain. That is itself a very useful signal for the brain to try to model in its decisions. So that was the kind of phenomenon we wanted to capture as an example of where our exteroceptive perception might be conditional on our interoceptive perception, linking back to this overall self-inference model, where the self-inference is literally the idea that you're building a model of how your own percepts, your own decisions, and your beliefs depend upon the contingencies between what your body is doing and the world. So to do this, we applied a modeling approach developed by Carl Friston, where I'm not going to really go into the details of this. It gets quite in the weeds. You can check out the bioRxiv preprint where all the math is there. Actually, all of the code for the model is available on our GitHub, so you can download it and run it. The basics of this model are that it uses a Markov decision process to model transitions between different discrete states. So those states might be like whether a mouse moves left or right in a maze, or whether the heart is beating or not. And the model has high-level policy priors, which dictate the link between the goals of the model, like getting the cheese at the end of the maze, and the sequence of actions and sensory states that are most likely to lead to those goals, versus the low-level transitions between the different states, which generate prediction errors in a kind of predictive coding-like scheme and create sensory outcomes in a probabilistic way. So the agent is an active inference agent, which is guessing about the causes of its sensations and then acting on the world so as to minimize the error on those sensations in a complete causal loop. The more important thing to understand is that what we did with this model is we created a little synthetic cardiac agent. So we created an agent that has hidden states that represent a heartbeat oscillating between three discrete states: an early diastole, a late diastole, and a cardiac contraction or systole phase. The agent, these states are controlled by a matrix which determines that the heartbeat will always go in a certain order. So the agent can't have a cardiac arrest essentially. And the model will oscillate between these different hidden cardiac states to create a heartbeat, and in doing so generate noisy sensory outcomes, which are the agent's beliefs about whether its heart has beaten or not. And the mapping between these hidden states and these inferred states is controlled by sensory precision in the kind of way you'd expect from a predictive processing perspective. Simultaneously, the agent has little exteroceptive states. For example, it can perceive that it's seeing some nice flowers or some threatening spiders. And these generate probabilistic beliefs about those states. And then the agent is equipped with high-level arousal policies which dictate the transition probabilities between these different interoceptive and exteroceptive states. What that means is the agent is equipped with a kind of James Lange-like theory of emotion, which means that it generally prefers to be unaroused when it's seeing nice things, and it prefers to be aroused when it's seeing scary spiders. And the difference between those is the agent selecting a policy where it either is relaxed and it transits between all three cardiac states before looping, or where the agent is aroused and it skips the middle diastole stage to have cardiac acceleration. Now, the great thing about having a model like this is we can do things that would not be considered typically ethical to do in a human subject. Like we can go in and lesion the interoceptive connections or the exteroceptive connections by changing the precision on those channels, and then observe how the agent's behavior changes in simulated experiments. We did this, for example, in some simulated oddball type experiments where the agent views a series of flower stimuli before being unexpectedly presented with a threat or spider stimulus. We can do this in a way that lets us track how the agent's inferences change over trials and also how the precisions change over trials. And because the agent generates a synthetic heartbeat, we can have a kind of measure of heart rate variability across the experiment. We did that in such a way, we did that in such a way as to generate a little experiment where we compared one agent that had fully functioning interoceptive and exteroceptive sensory channels or precisions, and a lesioned agent where we had gone in and ablated the ascending interoceptive information by setting those precisions effectively to 0. So you can see in these simulations where we run the simulation many times and take the average of its behavior, of the agent's behavior, in red is the healthy interoceptive agent, and in blue is the agent with the interoceptive lesion. And on the left, you have a kind of measure of the number of heartbeats on each individual trial of the simulation. And on the right, you have a plot of the agent's expected probability to encounter either the spider or the flower on the next trial. And the takeaway from this is that we see that both agents exhibit a cardiac acceleration. So when the spider is presented in the dotted line, there's an increase in heart rate. So both agents correctly select the aroused policy. But the lesion agent shows a blunting of this physiological, almost startle reflex-like response. And when we look at the fear expectations, we see that even though both agents dramatically revise their beliefs that they're about to encounter spiders after having encountered one, the lesion agent revises its belief to a lesser degree, so it's less sensitive to changes in the world. And interestingly, in the baseline period before we present any spider at all, the lesion agent is more likely to sort of hallucinate threats that don't exist. So this is a simple toy model that demonstrates how simply by linking the precision between an exteroceptive or visual channel and interoceptive channel, you get quite complex psychophysiological phenomenon. We also model the agent's uncertainty, measured as the entropy in each of its beliefs. I'm not going to go into this in great detail, but you can see here a range of different simulations where we adjust either the interoceptive precision or the exteroceptive precision. We can see that there's a kind of interaction. So the agent is sort of summing the confidences between the different channels, as you might expect a Puget-like Bayesian agent to do. And really nicely, for some of the work that people like Sarah and Manos have done, when we look at the uncertainty of the agent on each individual heartbeat across our cardiac cycles, we can see that there's a clear modulation of uncertainty by the heartbeat owing to that sensory attenuation. So that's kind of a quick overview, but that's the basics of the model. It allows us to pull apart different parameters. And we can even generate synthetic heart rate variability traces by looking at the agent's heartbeats and modeling them with kind of typical time frequency analysis. I won't really go into that here, but in the paper you can see that we generate different phenotypes of interoceptive inference, for example, a healthy agent or an agent who has a hyper-precise arousal prior or a hyper-precise visceral sensation. And we just show that by tweaking these different parameters of the model, you get very different physiological phenomenon, which in the future could be used to actually do a kind of interoceptive phenotyping of different patient groups. So I want to make sure I have enough time for the second part. So I'm sorry for the audience that's going through at breakneck speed. But
4 or 5 minutes?
Sorry?
4 or 5 minutes is okay?
Yeah, I can do it. So what we have is a model of interoceptive processing, but we don't only want to be able to model interoceptive beliefs and their confidences, we also want to have a way to accurately measure them. And that's something my lab has been working on quite a lot. I'm not going to go into a lot of the details here. I'm sure you'll hear about them in the other talks. There's a variety of existing ways to measure interoception, ranging from subjective report scales, heartbeat counting, heartbeat discrimination, and methods of constant stimuli. And there's a kind of a raging debate about the best way to do these methods. All of the existing methods have different trade-offs. In particular, a lot of the debate around the heartbeat counting task has focused on the role of subjective beliefs in the heart rate. There's a lot of experiments, for example, a classic one where they show that patients with pacemakers who are having their heart rates ramped by a huge range of something like 60 beats per minute, that the change in their heartbeat counting is more related to their beliefs about the situation rather than the actual physiological change in their heart rate. And that's something that's also been repeated in other experiments, showing that if you ask people to predict how much their heart rate will change, whether they're sitting or standing or in another position, that these predictions are more relevant for their interoceptive accuracy scores in a heartbeat counting task than their actual sensory information. Now, we don't, I'm going to kind of skip over this. There's also some other tasks we could talk about, but I don't want to run out of time. Basically, we can think about these tasks as trying to measure the ability to detect pure ascending cardiac sensory awareness, so the afferent signal of a single heartbeat. And in that context, it makes sense that you would try to exclude interoceptive beliefs about what my heart rate usually is. I'm going to skip over this too, sorry. But what we're trying to advocate for, and I think a lot of the field is really interested in, is moving from this kind of classical feed-forward account where it's all about detecting single heartbeats to the exclusion of beliefs, but instead about a kind of hierarchical Bayesian model where your interoception is a precision-weighted integral of, say, my prior belief about what my heart rate is, my interoceptive sensations that are ascending, and the overall metacognitive beliefs about interoception. To try to provide a measure for that, we've recently released a preprint describing our heart rate discrimination task. The basic idea is pretty simple. You listen silently to your heart rate for a 5-beat interval, and then you make a forced choice judgment about whether some auditory feedback is faster or slower than that heartbeat, and you rate your confidence. Behind the scenes, we use a psychophysical staircasing procedure to adaptively titrate the difference between that feedback and the true heart rate, so that over time we can converge on a threshold, which is your average ability to discriminate the heart rate. And what's great about that is it allows us to really carefully control the difficulty of the task while still having a sensitive measure of interoceptive belief. So here you can see how the task works. We have a subject with about a 60 BPM on average heart rate. The blue dots are correct trials and the red dots are incorrect trials. And what's happening is as we present feedback that are over or under the heart rate, the staircase algorithm is converging on the minimum difference that the participant can reliably discriminate. This allows us to estimate things like the signal theoretic bias, so that's the overall tendency to misperceive the heart rate as faster or slower than it truly is. We can have a very good estimate of the metacognition because the task allows us to gather about 150 trials or so in 20 minutes. And because of the staircasing procedure, we're able to push people around into very easy trials and very difficult trials, giving a very nice measure of their overall metacognitive ability. I kind of skipped over it. We've done some simulations that show that there can be some really problematic issues in estimating metacognition in a typical interoceptive context, and that's something that our task is kind of designed to solve. And we're still in the early days. Unfortunately, our pilot data collection was interrupted by COVID-19, but we've run some pilots in about 7 subjects so far, and we have 2 different methods of the task. One uses a classical in-up, in-down, and estimates the interoceptive threshold, which is the accuracy of the interoceptive belief. The other uses a Bayesian method. I'm not going to go into the details here, but essentially what it's doing is estimating both the threshold and the slope of a psychometric function for the heart rate discrimination choice. And that allows us to parameterize both the accuracy of the belief in the heart rate and its precision or uncertainty. And those are 2 parameters we think are going to be really important in future phenotyping studies. This is just some pilot data that you can see on the preprint showing that we can use hierarchical Bayesian models to estimate these slopes. We can express the uncertainty in those, for example, when a subject is not able to perform the task. That might be useful for discriminating interoceptors versus non-interoceptors. And our early pilot data, I think, show really nicely that the task can do a very fine job of controlling for difficulty. For example, in the in, up, down versus the psi, we can shift difficulty by about a full D prime, or about a half a point of D prime. I think really interestingly is our criterion values here. So we can see that most subjects have a negative criterion corresponding to a tendency to misperceive the heart as faster than it is. We think it's going to be really interesting to look at maybe patients with anxiety and see if they have a systematically more negative criterion. So we have a lot of ongoing work with that. We're doing a lot of work on the test-retest reliability. We're doing breath holding tasks to manipulate the heart rate. I didn't really get to go into it, but we have an exteroceptive control condition so that you can compare the delta in the 2 task parameters. And hopefully this summer we'll actually have some fMRI scanning from the task. So that's going to be I think, a really exciting development from our lab that's going to hopefully give us a more precise measure of the precision and accuracy of interoceptive belief. I think I'm out of time, so I'll stop there. I'm sorry for squeezing 2 talks into one. I have to say thank you to Manos, and thank you to our group, all of our postdocs and PhD students who worked on this project, and our funders. Thanks.
Thank you very much, Micah. Thanks. Right on time. And we do have the first question, which I will try to go through it. It's from David Moore, who says, how much do you think that there is a G factor of interoception? In other words, if a person is highly aware of heart rate, would they also be aware of other visceral sensations? Or is there a range of interoceptive signals that may independently vary in sensitivity?
That's a really interesting question. I think we don't know yet. There's a little bit of preliminary evidence, for example, from Sarah's lab, that shows some correlation between respiratory and cardiac interoception, but we don't have, really large-scale studies in all kinds of different populations to assess that. And I think we also still have a little bit of a measurement issue there. That being said, in metacognition itself, there is some evidence for a G factor in the ability to monitor different types of decisions. And part of our model, I think, is hypothesizing that a lot of the shared variance between different interoceptive and even exteroceptive channels is going to arise from the metacognitive level. So I think it's definitely possible that there's a G factor in interoception, but it's too early to say for sure.
Okay. Next question from Teresa Aurora. Your talk has focused a lot on heart rate. She's new in the field and was wondering if there are any data on appetite regulating hormones and the subjective feeling of hunger. And I know you're doing some interesting stuff on that.
Yeah, so we have, it's a really good question. And I think what's great about the hunger hormone literature is you can dive into it, and there's a huge amount of work. There's some beautiful papers just come out in, I think, Neuron and Nature Neuroscience earlier this year, doing optogenetics in the insula to see how hunger states relate to decisions and things. So there's a great literature there. I think it's still a little insulated from the cardiac respiratory literature. There's some stuff there, but not a lot. We do have a collaboration at Cambridge Psychiatry where we're looking at patients who've had prophylactic gastrectomy, which is really ablating a lot of those leptin and ghrelin-like hormones, and we're measuring other domains of interoception there to see if that disruption on the gastrovisceral channel will have a more domain general effect. So I think it's a really fruitful area, as long as you're willing to put balloons in people's stomachs sometime. Sorry.
Okay, next question from Laura Crucianelli from Stockholm, I believe.
Hi, Laura.
So she's thanking you for your great talk. She was wondering whether the model you presented in the first part of the talk does take into account also the noise associated with the heartbeat signal per se, such as skeletal muscle response, when you're trying to calculate the precision, or if the precision around certainty refers only to the subjective perception confidence of the heart signal.
That's a really nice pointed question. So the most important caveat about that model is it does not have biophysical realism. So it's really a toy model of trying to demonstrate what happens if you have a cyclic change in precision on an interoceptive channel linked to an exteroceptive. So, if you look at the synthetic heart rate variability traits, they're not anything that a human could live on. So that informs the second part of the answer, which is you anticipated exactly correctly that the precision there is not necessarily heart rate variability precision, although the agent does exhibit trial by trial heart rate variability. It's the precision of the mapping between the hidden cardiac or exteroceptive state and the belief. So it's more like the subjective belief precision than the physiological noise of the heart.
Okay. And I think with that, we can end your talk there. And thank you once more for taking part and giving this excellent paper. Very much appreciated.
Thanks, guys.
And
Thanks
I think we're ready to move on to our second speaker, which is a great pleasure to introduce a very dear friend and collaborator to some respect. And of course, a rising star in the field of interoception, Sarah Garfinkel, who's professor of neuroscience and psychiatry at the University of Sussex and the Brighton and Sussex Medical School. until the 31st of August, because after that, we're very excited that Sarah will be coming closer to us, joining the faculty at the Institute of Cognitive Neuroscience at UCL. Sarah, thank you very much for joining us, and we look forward to hearing your talk on interoception and fear memories.
Thank you, and thank you, Manos, for organising this symposium. It's a great pleasure to speak, although I wish I could be doing it in person. So today I'm going to talk to you about interoceptive signals and how they can selectively enhance fear memories. So when I was doing my postdoc in Michigan, I was working with individuals with post-traumatic stress disorder, and I was struck by these individuals who were stationed in Iraq and Afghanistan. And when they came back to America, they were coming back to a completely different context, which is a much safer context. And yet they still always said the same thing, which is the world is an unsafe place. Even if they were in Ann Arbor, which was a sleepy, a very safe place, they still had this feeling that the context and the world was unsafe. And this made me investigate this empirically. So how can individuals with PTSD potentially not use context in a way to facilitate safety memories? So is there something altered about their use of the external context? And could this be driving fear learning and fear memories? And this made me interested about the role of the internal context. So what is it about signals from the body, for example, the heart, which can interact with the brain and shape fear? And could this be altered in individuals with PTSD? So when we think of the senses, we tend to think of vision, hearing, sight, sound, taste and smell. But these are actually classified as exteroceptive senses. That is, they tell us something about the outside world. And in contrast, we have proprioception, which is sensing the position of the body in space. And we also have interoception, which is sensing internal bodily sensations. And Michael already did a great job of defining interoception. And he highlighted a model which I've now expanded on. So for me, all of these different dimensions are interoception. And actually, it's really very aligned with how Micah is also seeing interoception, where you have the afferent signal itself, and this can be sensed neurally. You have the preconscious impact of the afferent signal, so how signals from the body can change stimulus processing. You have interoceptive accuracy, and I'm so excited to see Micah's new way of testing it, which looks really exciting. You have belief about how good you are, metacognitive measures, and higher order belief and attribution measures. And we can measure these in individuals to see how interoceptive profiles differ. And I'm going to speak to you a little bit today about the preconscious impact of the afferent signal. So every time the heart beats, it sends a signal to the brain. Micah again highlighted the pulsatile nature of this signal in the brain. And so it's long been known since the 1970s that you can time stimulus presentation to very precise points of the cardiac cycle, either at T wave, which for simplicity I'm going to call cardiac systole, which is during the cardiac ejection period when the heart is actually beating and the heart-brain channel is active. Or you can present stimuli at R wave, which is late diastole, which is in between heartbeats. And it's at this point that the heart-brain channel is quiet. And if you present stimuli at very specific points in the cardiac cycle, you can see how this active heart-brain channel alters stimulus processing. And it's long been known since the 1970s that these cardiac afferent signals can have inhibitory properties, where if you present sensory stimuli, for example, electric shocks, and look at EMG response or startle response and other things, you can see that there's a dampening down or reduction in the response 200 to 400 milliseconds after the R wave, which is coinciding with this active heart-brain channel. So this was seen as the inhibitory property of the heart. I extended this into the domain of memory, where I presented words at very specific points in the cardiac cycle. And I was able to show that using an incidental memory test, that if you test people's memory for words, they're more likely to forget them if they saw them exactly when their heart was beating relative to in between heartbeats. But this made me wonder, because at the moment, it was said that these heartbeats were always inhibitory. And because when the heart-brain channel is active, what the heart is actually signaling to the brain is a degree of cardiovascular arousal. And I wondered that if we could selectively time fear stimuli to this precise point, could fear processing actually be facilitated and not inhibited? So I selectively presented either fear or neutral faces at systole or diastole and got people to make intensity ratings. And I was able to show that contrary to what you normally see, fear stimuli were judged as more intense when the heart was beating at systole relative to diastole. And the reverse effect was shown for neutral items, which was in line with this inhibitory effect of the heart, which fear was not showing. And if we see at the level of brain, because individuals are scanned at the same time, that signal in the amygdala was modulated by both the heart and emotion and tracked the cardiac change in emotion ratings with higher amygdala activity for fear stimuli as systole relative diastole, with the reverse being true for neutral items. And once you've set up a paradigm like this and shown how cardiac afferent signals can alter emotion processing, you can take it into patients to see how interoceptive influences on emotion may differ in different patient populations. So this was in 386 patients. And I don't want to spend too long on this data because I just want to set the scene for the experiment I really want to talk to you about today. But essentially, this was using the same paradigm, presenting neutral faces and fear faces at systole relative to diastole. And this is looking at the cardiac modulation of neutral faces specifically. And you can see this dampening down of neutral intensity ratings in basically every different clinical condition, with the exception of anxiety and schizophrenia, where you were not seeing this effect. And this contrasted to fear, where in healthy controls, we replicated this small but significant increase in intensity ratings for stimuli, fear stimuli, systole relative to diastole. This increase was further augmented in anxiety disorders, showing individuals with generalized anxiety, the cardiac afferent signals further boosted fear processing. There was no cardiac modulation of fear in all of these different conditions, and the opposite effect, reduction in intensity, was observed for fear at cardiac systole in individuals with schizophrenia. So this now leads me on to the experiment that I want to talk to you about today, which is a memory experiment. So can we take this into the domain of memory, which is really inspired by my PTSD work, although these individuals are all healthy, mildly anxious controls. So my hypotheses were cardiac afferent signals will selectively facilitate fear learning and fear memory, so extending this fear facilitation effect into the domain of memory. The second hypothesis was that this cardiac facilitation effect will be augmented by anxiety. And then the third hypothesis was the heart will serve as an internal context to facilitate memory recall. And this was really inspired by work which has typically manipulated external context. So when you have an external context, it can facilitate memories learnt in that context, and changing the external context can disrupt those memories. And I wondered if we see the heart as an internal context, if we alter the cardiac signals, can we get an alteration in the memory? I'll explain that later, but basically it's looking at the heart as a context. And now it's so hard to explain this experiment. It's like the most complicated experiment. So to do it actually adds an extra channel, but hopefully you will bear with me. It's basically a 2-day fear conditioning experiment. So on day one, we presented different condition stimuli, which were different neutral shapes. And we presented 2 of them with an electric shock 70% of the time. This set them up to be CS pluses. And we presented 2 different ones with no electric shock, setting them up to be CS minuses. 1 CS plus and 1 CS minus was presented at cardiac systole, whereas 1 CS plus and 1 CS minus was presented at cardiac diastole. This happened to establish fear conditioning and condition learning to the CS plus versus CS minus, and to look at the cardiac effects. Then immediately following this phase, we individuals underwent fear extinction. And this is where it now splits into a between participants manipulation, where for half the participants, they were extinguished using the same cardiac contingencies. So those stimuli that were presented at systole were still presented at systole, and those stimuli presented at diastole were still presented at diastole. And for the other half of the participants, the cardiac contingencies swapped. So stimuli previously presented at systole were now at diastole, and diastole now at systole. Because this was the extinction phase, there were no electric shocks presented during this phase. And then the next day, participants returned. They were given 2 random electric shocks just to reinstate fear. And they were again shown the 4 conditioned stimuli, but with no cardiac time locking and no electric shocks, to look at the nature of fear memories the next day. Okay, I'll explain it more as we go along. So again, this is the fear conditioning session where we have a CS plus and a CS minus, a systole and diastole, and we're looking at skin conductance response as our measure of fear. And for early fear conditioning, in the first half of the experiment, and this really blew my mind to get this so significant, was that you were not seeing an effect of CS plus versus CS minus. What dominated initially was the effect of the heart, with heightened skin conductance response for stimuli at systole relative to diastole, irrespective of whether there was a CS plus or a CS minus. So stimuli associated with these cardiac afferent signals were generating a fear response, and that was overshadowing their ability to differentiate what was a CS plus and what was a CS minus. So initially, fear was tagged to the heart. And we found that this heightened effect of fear as a function of cardiac systole was driven by those individuals with high anxiety. So in those individuals with high anxiety, they had a heightened skin conductance response for stimuli at systole, and you weren't seeing that in the low anxious individuals. Showing this same medium split analysis as a continuous measure where anxiety now is a continuous variable and each dot is a person, you can see this is skin conductance response at systole relative to diastole, and there's a heightened skin conductance response as a function of higher trait anxiety for things presented on the heart relative to in between heartbeats. We found that shock expectancy ratings did not differ as a function of systole and diastole, but there was actually a change in pleasantness. So for stimuli which were a CS minus presented at systole, they were showing a reduction in pleasantness. Now for our extinction manipulation, where we had then established fear conditioning with people showing a differential skin conductance response to CS plus relative to CS minus, irrespective of the heart. And once that was established, participants then underwent fear extinction. And so now no electric shocks were presented, and they were, for half the participants, the different stimuli were presented on the same heart contingencies. So stimuli at systole were still at systole, stimuli at diastole were still at diastole, and for half of them, they switched. And extinction was effective. I won't go into the details of that. And then the next day, individuals returned to the lab. They were given 2 random electric shocks to reinstate fear. And then they saw the 4 stimuli again, but this time they were not time locked to the cardiac cycle. So we could see the effects of yesterday's manipulations on fear memories. And what we saw initially was this main effect of the heart, with a higher fear response to stimuli previously seen at systole relative to diastole, showing that the stimuli associated with these cardiac afferent signals were generating a higher fear response, irrespective of whether they had been previously paired with a shock. So this main effect of the heart prevailed. And then regarding the analysis about whether there's a heart can serve as a context, in those individuals who were extinguished using the same cardiac contingencies, they showed the classic reinstatement response the next day. So they had a higher fear response to stimuli previously associated with shock relative to no shock, whereas those individuals who had the switch in cardiac contingencies during extinction show a disruption of this classic effect of this differential fear to CS plus relative to CS minus. So to summarize and conclude, we hypothesized that cardiac afferent signals would facilitate fear learning and fear memory. And yes, we did observe that. So during day 1, there was a heightened fear response initially to stimuli presented at cardiac systole relative to diastole. They were generating the fear responses, and this initially overshadows people's capacity to have differential fear response to stimuli predicting shock relative to no shock. And this did actually extend at the group level to day 2, where when seeing these conditioned stimuli, again, people had a higher fear response, skin conductance response to stimuli previously shown at systole relative to diastole. So again, these stimuli coincident with these cardiac afferent signals were boosting fear. We hypothesized that this cardiac facilitation effect would be augmented by anxiety. And we did see this during learning. So this capacity for cardiac afferent signals to boost fear was shown to be augmented in high anxious individuals. But we only actually saw this effect during day 1. I didn't show the data for day 2 regarding anxiety, but actually the next day it was not significant. But this is not a clinical population. And then we also hypothesized that the heart will serve as an internal context to facilitate memory recall. And it's true that we saw a preservation of the differential fear response to CS plus relative to CS minus only in the group that were both conditioned and extinguished using the same cardiac contingencies, whereas those individuals that switched them showed a disruption of this memory the next day. And then finally, it makes me think about implications for conditions such as PTSD. So could there be something about individuals with PTSD who have a heightened heart-brain channel or heightened capacity for cardiovascular arousal in the brain to facilitate fear? And could this potentially be driving, to some extent, the fear memories? So I'd like to thank everyone in my lab who helped, and my collaborators, and the ERC who funded this research, and I'm very grateful to. And that's it. Thank you.
Thank you very much, Sarah. Thanks for a wonderful talk. And we do have a first question, which is a manipulation check question. In the conditioning task, trait anxiety was not associated with differences in heart rate in general. And the reason why this person is asking is because they're interested to know if it is driven by when a CS is presented or whether by differences in heart rate in general.
So when I remember that when we added heart rate into the analysis as a covariate, it did not get rid of the result. So it was not driven by heart rate. The effect of anxiety was maintained when that variable was controlled for.
And I guess the follow-up question would be whether you think that some of these findings can be explained by the fact that individuals high in anxiety may also have greater awareness of their body, as suggested by some tasks.
Well, this is a preconscious effect, though. So people aren't aware that these stimuli are presenting on or off their heartbeats. It's an unconscious manipulation. So these signals are boosting fear in these high anxious individuals. Because my way of seeing interoception is all of these different levels, they are related to some extent. So yes, individuals with anxiety may also be more aware of their heart, and that may speak to, again, an augmentation of this heart-brain channel. So I think it's part of comprehensively understanding anxious individuals, the fact that this channel may be boosted may increase their awareness of it, increase the ability of it to shape stimulus processing. I personally don't necessarily think that anxiety is associated with it is associated with higher awareness if they're highly aroused and the heart is beating strong and fast. But I think anxiety is also associated with a lot of error about the body as well. So it's also complicated delineating that. But yeah, hopefully that answers it with nuance.
Excellent. Then we have some congratulations from Berde Herbert. She's thanking you for your great talk and the elegant experiment. Michael Gabler, great results and talk. Did you analyze at which cardiac phase stimuli were presented during the reinstatement?
No, because we just presented them randomly, but I know that he does really elegant work with these circular analyses to then see if there's an effect. So we didn't do that because we weren't powered to. We didn't have quite enough trials, we thought, to necessarily do a circular analysis. We also wanted to take out the cardiac effects on day 2 because there were already so many conditions with systole, diastole, switch contingencies, not switch contingencies. We didn't want to add an extra layer of contingencies the next day as well. And we think our results fell out quite nicely when we smoothed over that variable. But I think he has a good point, and I do love his circular analysis based approach. So we could retrospectively analyze those data to see if there's an effect.
I think most of your stimuli would fall in diastole, as this is the larger phase of the cardiac cycle.
100% is also true. Yeah.
Okay. Roy Salomon is thanking you. Ruben, hi Sarah, great talk, fascinating last study. I was wondering if you are aware of these TDCS studies showing enhanced recall of information retrieval when encoding and retrieval occurred at similar stimulation frequencies compared to when encoding and retrieval occurred at different stimulation frequencies. And if you think that the similar principle would be at play here as well, but instantiated by physiological states. So I guess, you think about the congruency between physiological states, either spontaneous fluctuation like the ones you did, or by means of brain stimulation.
I actually didn't know of those experiments. I'm so pleased you brought them to my attention. Was that Ruben that did that? Thank you, Ruben.
I think Ruben asked, but I'm not sure that he was the one who did the study.
Okay, well, I appreciate him bringing it to my attention, and it's absolutely in line with, about this internal context and getting states in brain states or body states in a similar state to facilitate. So it's very much in line with the thinking, and thank you for, bringing it to my attention.
Okay. JL, he or she is curious about something you showed before. Why would you think the signals boost fear in anxiety and do the opposite in schizophrenia, as you showed before? So why that difference between conditions?
So I think it's a great question. And for me, this is the biggest thing that I don't yet understand. So first of all, I think it's easier to understand from the perspective of anxiety that when the heart is in active communication with the brain, it's signaling to the brain a degree of cardiovascular arousal. So when you're in a high anxious state, then that and high cardiovascularly aroused state, then that is a boosted signal. So it's sort of a mechanistic approach, an insight into a system in the brain. And it makes sense that cardiovascular arousal state, high cardiovascular arousal state, selectively boosts fear, which is a type of processing which is compatible with that type of brain state. And it makes sense to me that would be facilitated in anxiety, and the modulation of the heart with the brain during different states can act like kind of carrier waves. So you have amygdala activation that can occur with the beating of the heart, and that heightening of that signal can boost anxiety. So it makes sense to me why that might occur. What is telling me about schizophrenia is there's something different about the way that cardiovascular arousal is processed in the brain, and it's not having that boosted effect. And what you normally see about the heart, and that's how I opened out my talk, was that these cardiac signals are normally inhibitory. I normally interfere with stimulus processing. And what schizophrenia is showing is the inhibitory effect. So it's an extension of that inhibitory effect into a domain when you wouldn't expect it. So the inhibition isn't so surprising because things tend to be inhibited at the heart, but it seems that fear is something that can get boosted or can be impervious to this deleterious effect of the heart. And schizophrenia is just showing this generalized inhibitory effect. And I don't have an answer necessarily for why.
Okay, and one last question. Thank you, Sarah. Really interesting talk. Do you think that interoceptive accuracy or other dimension would moderate the effect of systole on fear memory? As in, do you think that an unconscious or conscious awareness of heartbeats might attenuate the facilitative effect of systole or enhance it?
Yeah. So it's a great question, and we do often see effects of interoceptive accuracy boosting these cardiac modulation effects. So if that channel is clearer, then we do see quite often an enhancement or a reduction of that effect, depending on what the mechanism or type of processing we're looking at specifically is. So the effects with this one were very, very small and not striking at all. But I do wonder if we, for example, used other tests that might be better at looking at interoceptive accuracy, maybe Micah's great new test.
So you had interoceptive accuracy as a measure.
We do. And actually putting it in and controlling for, so when we put in interoceptive accuracy as a covariate, then it was more likely to bring out the conditioning effect of differential skin conductance of CS plus versus CS minus when you control for interoceptive accuracy. Mm-hmm. So it did have an effect, and controlling for that effect made the conditioning effect more likely to occur rather than the fear of the heart effect dominating.
Yes. We have been using the same method and approach in our studies, time blocking things at different phases of the cardiac cycle, and by and large, we don't find that the effect of the baroreceptor firing is modulated by levels of interoceptive accuracy. But in a series of studies that we recently did, where we tried to look more carefully on the role of interoception for social cognition, this is the only case where you combine a more mechanistic manipulation like time blocking things at different phases of the cardiac cycle with interoceptive accuracy. You do find an effect whereby people with different levels of interoceptive accuracy, they may behave differently depending on when things happen in the cardiac cycle. And I think that also fits with, ideas about the role of awareness or consciousness in general, that it is very much about other people rather than about your own self. So if you like, in a rather solipsistic, although nothing is solipsistic, but in a rather solipsistic context where you just have to respond individually to a fearful stimulus, levels of interoception or interoceptive accuracy may not matter. But when you have to communicate that to someone else or share the information or form a judgment about someone else's emotion, this ability to be accurate or not may matter.
Yeah. I totally agree with that. That's interesting.
Okay, I think we're doing very well with timing. I think we have a break. We can zoom out or Skype out for a while and then come back in about 15 minutes to listen to the talk by Catherine Talon-Baudry and then by Antonio Damasio. Thank you very much, both Micah and Sarah. I hope you can stay until the end, but if not, it's been a great pleasure having you. And thanks all of you for tuning in, and we expect to see you back in about 15 minutes. Bye for now.